Sorry about that, everyone. Welcome back. Welcome to all the new attendees. Today we have here Dr. Michal Tal, who is an instructor at Stanford University School of Medicine. She studies host pathogen interaction with a focus on immune clearance and diversity of immune response to tick-borne diseases. She received her PhD at Yale University in immunology under the mentorship of Akiko Iwasaki, researching immune responses to RNA viruses and how they are impacted by processes such as aging. She then did her postdoctoral training in the laboratory of Irving Westman at Stanford, where she's currently an instructor leading the infectious diseases team and studying immunomodulatory mechanisms which impact human clearance of infectious diseases. She received a Yale Gershon Fellowship, NIH and F30 and F31 and F33 pre and postdoctoral fellowship, and was selected to give inaugural Janeway early career lecture at Yale University. So thank you, Michal, for being here. Sorry everyone for the delay, and if you have your slides, you should be able to share them now.
Okay. I'm so sorry, everybody, for getting the timing wrong. I thought we were starting an hour from now. So I'm just going to go ahead and dive right in. Today, I wanted to talk to you guys about my research, which has been about a self-recognition axis called CD47. And I'm just going to go into full screen here.
So guys, sorry I didn't mention, but if you have questions, you can just post them in the chat while Michal is talking, and then we'll read them in the end and she'll be able to answer. Okay, thank you, Michal.
For sure. Okay, so the important thing here is that your immune system needs to be able to recognize which cells belong to you and which cells are foreign. And so you have all sorts of mechanisms to maintain what we call self-recognition, where your macrophages and other immune cells, they can tell the difference between cells that are supposed to be there and cells that are not. And one of the reasons that this is really important is that our body is often under attack from different pathogens that are trying to get in and trying to replicate, and the immune system is battling them. But in battling these pathogens, often the immune system itself also causes disease. I think this is something that we're now seeing very clearly with COVID, where in late stages, it's actually kind of the immune system gone wrong or dysregulated, which is directly causing disease as much as the pathogen is directly. So we have this axis, and one of the things that becomes really important in different diseases is to be able to figure out who is causing disease. Is the pathogen causing the disease, or is the immune system causing the disease? And this makes for very different modes of treatment. And the immune cells that we're going to talk about here for the rest of the talk are a type of immune cells that we call phagocytes, and we're going to focus specifically on macrophages, but neutrophils are also very important phagocytes. And what phagocytes can do is they can do a lot of eating. So I wanted to show you guys, this is a video from the NIH, which I thought was particularly cool. You can watch this macrophage in the middle, eat a bunch of red blood cells. And here it is. It kind of grabbed them, and now it's engulfing them and bringing them inside. So that's what phagocytes can do. And here, just gonna, okay, here we go. So we are going to talk about these two very particular proteins that mediate an important part of this self-recognition and how the macrophages decide what they are going to clear and what not to clear. And these two proteins are called CD47 and SIRPa. So CD47 is the ligand. This is what is seen. This is displayed by every single cell in your body to say, I'm okay, don't eat me. And so we call it a marker of self. And SIRPα is an inhibitory receptor, which can see that signal. And when it sees that signal, it can tell macrophages not to eat. And this work was really spearheaded by Irv Weissman, who I work with. And so we made this joke about, 47 just helping everything eat more, blocking 47. So here you're seeing how, a schematic of how we would think about this, where CD47 is on the target cell and SIRPα is on the macrophage, and there's going to be this interaction between them, which is going to decide whether or not the macrophage is able to eat or is inhibited from eating. And what I wanted to make the point about right at the beginning, we'll talk about this at the end, but I wanted to make the point right at the beginning that the inhibitory receptor SIRPα is extremely diverse in the population. So for those of us that are on the call, likely we all have very different looking SIRPα proteins. And here I just drew them in different colors for you, but it's much more nuanced than that. Obviously, it's a mutation here or there within the protein that would be different. And the reason that this is very important is because CD47 actually, all of our CD47 probably looks very, very similar. And so it becomes very surprising that we all have very different looking SIRPα. And it becomes very interesting for us in terms of what that means for what SIRPα does, what that means for our diversity as a population. And the reason that we're in general very interested in diverse proteins is that your immune system knows the power of diversity very much. Your immune system, I mean, we have as a species evolved to have diversity in our immune systems, because otherwise there could be a bug that would come along that would kill all of us. By having diversity in our immune systems and by each of us responding differently to different pathogens, there's a sort of species level defense in that, that no one bug could get us all in the exact same way. So what does CD47 do? What is the impact of SIRPα diversity? These are the things that we're going to talk about. And so to understand what CD47 does, I want to start off with the history of CD47, which we really started to understand only in the year 2000. In this paper, where it was defined that CD47 is very high on young red blood cells. And over time, as red blood cells have lost their nucleus, they lose CD47. And this loss of CD47 actually ends up as a molecular clock, which tells your macrophages, okay, this red blood cell has been around too long. This one needs to get eaten and cleared. And this is how you maintain your turnover of red blood cells. So in the Weissman lab, one of the things that Irv really spearheaded is from defining this as how we clear our hematopoietic stem cells and many other immune cells in the body. He found this, and many people working in the lab found this to be CD47 was getting overexpressed by every cancer cell type that we tested. And they started working on using this blocking CD47 in lots of different types of cancers in combination with other cancer therapies. And this actually turned out to be a really, really, really important way to go about attacking the way that these cancer cells were protecting themselves from getting eaten, and has come out to be really exciting in different human trials, and spun out as a company, CD47 Inc., which was then acquired by Gilead. And all this time that I've been in the lab, my question has really been, okay, this is clearly extremely important in cancer. What does it mean for infection? And so we come back to this picture where I'm showing you that tumor cells use CD47 to hide from getting eaten by macrophages, by binding to their SIRPα. And what I wanted to understand is, do infected cells do that same thing? And is it possible that bacteria and other pathogens do that too by mimicking our CD47? And so we saw that infected cells, they really broadly upregulate CD47. I'll show you guys a little bit about that. But one of the things that we wanted to understand is, do they do that on purpose? Does the pathogen get the cell to upregulate CD47? Or is the host getting the cell to upregulate CD47? So we're going to take a look at that. And this is actually a paper that we published pretty recently. And on the left, I wanted to start off with all of the names of the authors on the paper, because this was such an incredible team effort. So many different groups and people really helped us get this done and look at this in many, many different infections. And in the picture on the left, you're seeing the 3 first authors. We had 3 co-first authors and 2 last authors. So that's in the picture in order. You see Laughing Bear, myself, and Laura. And then on the right, you're seeing, and then next to us, you're seeing Irv Weissman and Kim Hassenkrug. And in the middle picture, you're also seeing me with 2 of the graduate students that were working with me at the time, Laughing Bear and Phoebe. And on the right, this is the other co-author, Laura Myers, who I worked very closely with, and Kim Hassenkrug Saab at NIH. And the reason that I show this is science. Here you're seeing us on Zoom, even when our phones aren't, when our audio isn't working and we have to be on our phones. I think science, so much of science is who you're working with, and working with really great people. And so I just wanted to acknowledge them. This is the first figure in that paper. And here we show that very diverse infections lead to this upregulation of CD47 on infected cells. But one of the important things I wanted you to get from seeing this figure and from seeing the different ones is that the magnitude increase of CD47 is rather low. Like, this isn't, we don't see tenfold increases. We see a fairly consistent 2-fold increase, sometimes even less. What was really intriguing to us about CD47 upregulation wasn't the magnitude that it's increased, it was the consistency with which it's increased in infected cells from very different pathogens. So we found something that was just across the board, seen in so many different infections, even though it's not increased to a very big height. And interestingly, as we were getting this paper through review, we added in SARS-CoV-2 from a data set that was publicly available. And that was really exciting that we were able to see that. And off of additional data sets that have been available since then, we can see that CD47 is indeed upregulated on infected cells and correlates with, and it's most upregulated when the viral titer is high. So that was really interesting and interesting. And so what we really wanted to understand is, if all of these different infections can upregulate CD47, does that mean that that they've all evolved this way of modulating the host? Or does the host have a default pathway which causes the upregulation of this protein? And so in order to do this, we trick the immune response into thinking there's a pathogen when there's not. We've just put in, some synthetic molecule and fluorescently labeled so we can see which cells took it up. And what we see is that if we separate on the cells that have taken this up and that it's triggering different pathogen recognition receptors, so they think that they're infected or they think that they're sensing an infection, what we see is that in both cases, it leads to this height of CD47 upregulation that turns out to just be a default host pathway. It means that when the cell thinks that it's infected or thinks that it's seeing infection, it turns on CD47. But this brings up a really important question, which is, why would it do that? And why would macrophages get instructed to ignore infected cells? That seems counterintuitive. That doesn't really seem to make sense unless you're thinking about the fact that, unless you're thinking about this part where the immune system itself can cause disease. And so if you're thinking about how the immune system, if it's doing this, if it's going so strong and doing this incredible job fighting the pathogen can't replicate, the pathogen is almost gone, it can be so strong or even unorchestrated. Here it looks like they're all acting together, and that would normally be good if that was true. Sometimes you've got some acting this way and some acting that way. But if you let this go too far, this can actually cause a tremendous amount of disease. This could even kill you. So it's not the kind of thing where inflammation needs to be tightly regulated. Too much is bad, too little is bad. You really want to hit this kind of sweet spot. And so what we realized is, and this is showing, of course, too little is also bad, because then you just let the pathogen be out of control. But so what we really realized is that CD47 blockade has this incredible potential because CD47 itself is a checkpoint. So CD47 is one of these brakes on the immune system that's telling it, don't go too far. You know, we know there's an infection here, but keep it tame. And so one of the things that we tried to see is if we blocked CD47, could we ramp up response to an infection? Could we unleash a little bit more inflammation to let this go? This is a paper by our collaborators, to which we contributed this interesting piece in HIV infection, where what we were able to show is that by blocking CD47, we were actually able to reduce HIV viral load and also rescue this CD4-CD8 T cell ratio, which gets really disturbed into HIV infection. So we'd come back to this point where, okay, so now we know that CD47 is this host checkpoint. But we also knew this other piece, which is that this particular type of viruses, these pox viruses, they encode a CD47 mimic. And that's really important because we've actually been evolving with pox viruses for a very long time. And they could very well have been driving this diversity that we have on SIRPα. And so we were interested what other pathogens might have been doing this. And if you look at the pox virus CD47 mimic, it very much looks like it's been stolen from mammals. And it looks like very much like the host CD47 molecule. So what we had in our arsenal at this point is we had reagents that we could use to bind CD47 and not block it, to block CD47. And now we were able to make this reagent where we could block the entirety of the CD47 SIRPα interaction. And the way we were able to do that is that the crystal structure was known between how CD47 and SIRPα bind. And so this is a paper that was published several years back. And what they showed is that if you mutate different residues of SIRPα that are known to bind to CD47, you can actually increase the affinity tremendously. So what we were able to do is make this very high affinity molecule that this engineered protein that can bind CD47 50,000 times tighter than your regular SIRPα can. And the other thing that I'm showing you with the red box at the top right here is that SIRPα on different people, these are just two of the most common mutations that are seen in people. So this is just looking at two variants, but even between these two variants, you see that they differed in how tightly they could bind to CD47. There was a twofold difference there. So there's some really important differences between us based on how our SIRPα looks and how it interacts with CD47. So we use this probe to look for pathogens that were mimicking CD47. And here's one that we found. This is on tuberculosis. And you can see that it's, even within the mycobacterium that are making a colony, you can see that it's on just a few of them here and there. And you can see that if you infect CD47 knockout mice with tuberculosis, they actually survive better than the ones that had CD47. Another bacteria that turned up in our screen, and now we've entered into unpublished data, but I thought this was really interesting to share with you guys, and hopefully something that we'll publish very soon, is that we find this mimicry of CD47 on Borrelia burgdorferi. And this is the bacteria that causes Lyme disease. And so we got very interested in terms of what this means for immune clearance. And what this means based on what your SIRPα looks like, how much this CD47 mimic is going to impact your macrophage's ability to clear this pathogen. So we wanted to see if blocking the CD47 mimic on Borrelia can impact clearance. So here's that macrophage that I have behind me. This is what it looked like if you're looking at that whole macrophage, going down in and finding this layer where we can see that whole Borrelia being eaten by the macrophage. And so this is that layer that we look at a lot. And what's happening here is that macrophages will actually extend a process and they will wrap that Borrelia like a lasso and pull it in. So this is very different than the way that they would eat round bugs or yeast bacteria that are round. They would eat them in a very different way than one that's really long and spirally, like a Borrelia spirochete. So how do we measure phagocytosis? One thing that we can do is we can have our bacteria in green, and then we can stain them with a dye that is acid sensitive. So when it hits that acidic macrophage stomach that we call the lysosome, it's going to turn this red. And so what's that going to look like? If we watch a macrophage eating, you're going to see here it's struggling with this Borrelia, struggles, grabs it, and it turns red. Now that the macrophage is red, we know that it's eaten that bacteria. And so what we can do is we can watch a whole field of macrophages, and we can watch them turn red, and every time they turn red, we know they've eaten. And so if we watch them go, we'll see that some of them are getting really excited and moving all around and really stretching and eating. And some of them aren't doing that much. And these differences between them have been something very interesting to us. And also you can see some of them are just working so hard. Some of them are even working so hard that they've eaten so much that they died. I'll show you one of those close up. So here's a close up of one of those. And we'll just look at it in action as this macrophage. Actually, I think it's gonna, we'll see it go off in a second. If you watch this one closely, this one that's eating, it's gonna work really, really hard. It's gonna grab and stretch and try to eat lots of different stuff. And then it's actually gonna do that so much that it's gonna die. And that's what happened just right there. So this macrophage actually got overactivated. And so part of what we're doing actually becomes this, like, how to train your macrophage and how to get it to be the best possible bacteria eater that it can be without pushing it so far that it just gets overactivated and dies, because obviously we don't want that either. So the other thing I wanted to point out from watching these is that there's this very clear macrophage diversity. You've been noticing it in the other ones. It's going to be really obvious here that the macrophages that turn red look one way, and the macrophages that don't turn red, they look another way. And you're going to see that these macrophages that are turning red and doing all the eating, they're very spindly, they've got all these processes out, they're getting thin, they're moving everywhere. And the macrophages that aren't really eating, they're just, you know, staying around and not really moving around. And so there's these important differences between these macrophages. And the reason that we think that this is so important is that different people respond differently to Borrelia burgdorferi, which causes Lyme disease. 90% of people can clear it with antibiotics, and they go on to be fine, and they, you know, hardly even remember that they've had it. Whereas you have this 10% that even when treated early with antibiotics, it's not going to be that simple, and it can actually have extremely devastating consequences of having had this infection, and it doesn't clear that easy, and the symptoms don't go away that easily. And so we've found that I think it's really important to really dive into these diversities in the immune system and see how different cells are looking at this, and how their genetics is also, how there's these different features of the immune response that also impact how we clear these bacteria. And so this is looking with that coating that we have, that CV1 engineered protein that we made. If we use that to get macrophages to eat better, you can see that some macrophages will eat much better, and some macrophages not so much, not really impacted by it. And we find that very interesting, this difference between different people. And here I'm showing you mice. There, we infected them with Borrelia burgdorferi, and we infected them with either a little bit, enough that they could clear it. Here, let me go back 1 second. So here we infect them with just a little bit and enough that the Borrelia can clear it. And here we infect them with a lot, and so much that they really can't clear it. And then we keep that same dose, but we cover it in this engineered molecule, CV1G4, or just in an IgG4 control. And what you can see is that over time, you can see that having coated it in CV1G4 was actually helping the mice to clear this bug. And if we look at this other type of antibody, which means, you know, that they really saw something for a while, you can see that 3 of the mice never even, when coated with CV1G4, they never even developed, like the infection never even really took hold. So we find that to be very interesting and very promising, that there's this important CD47 mimicry on these bugs that's really important for establishing the infection. And so this is the last thing that I wanted to tell you guys about, which is, as I mentioned before, your SIRPα is one of the most diverse immune proteins that you have. So it's actually the 3rd most diverse immune protein that you have. And this is a story that I've had a lot of fun working with. Here's Phoebe and Hannah and Amira, who work with me, and who work together with me as collaborators. And what I wanted to show you here is that this is, if you were looking at the genes that we have as humans, and you were just looking at the entire human population, and we colored for each different mutation that appears in the human population, you gave it a color depending on what type of mutation it is, and you gave it its height based on how many people with what percentage of the human population has this mutation. So then what you'll see is that in CD47, you see that there's a few sporadic mutations. Only a small percentage of the population has them. If you look at this part of SIRPα, you can see that there are tremendous diversities, and some of them are hugely frequent in the population. So a huge fraction of the population actually has these mutations. And interestingly, this blue bar codes for the amino acids that make this outermost region of SIRPα, that one that touches CD47, the one that needs to interact with it. So it is so incredibly interesting that we've been causing a lot of diversity there, right at the point where SIRPα needs to see CD47, and I'm showing you that CD47 is broadly the same across humans. So why is SIRPα changing so much at this site? Well, we think that it's being driven by these different pathogens that have been mimicking CD47. And so different people with the different ethnicities that have been exposed to these different pathogens, their SIRPα has been under pressure to try to tell the difference between their own CD47 and all these mimic CD47s. And that's what we think is happening there. And to put this into the terms that I just kind of want to end with, so what we think is going on here is that pathogen mimicry of CD47 has been leading to a lot of diversity in SIRPα. We think that there are very important differences between macrophage types and between macrophages from different individuals, which impact their ability to clear Borrelia. And we think that Borrelia have a protein that can mimic, we very much think and have a lot of evidence to support that Borrelia have a protein that can mimic CD47 as a don't eat me signal and bind to SIRPα in order to evade our immune clearance. And so I've talked to you guys a little bit today about 3 of the things that I work on in the lab and that the people who work with me work on, which is how CD47 is very important in infection, the surprising diversity in the human population, and that there's been pathogen mimicry of CD47. I also work on atypical antibody responses in both Lyme disease and COVID-19 and see some very interesting comparisons there. A lot of this work has been a huge team effort by a lot of different people. So I work within our Weissman's lab. He's incredibly supportive. I also have a group of my own, and so everybody with a little box next to them actually works directly with me. And then underneath that, you see other lab mates who really were tremendously helpful on the data that I showed in this presentation. Balanzaro, who's now a professor at UCSF, but used to be in the lab, Amira Barkal, Rachel Brewer, Benson, and Tasia, all super helpful. And within my group, there was work presented here from Phoebe, Laughing Bear, Paige, Sarah, and Maya. And then we also have a lot of fabulous collaborators. I showed you a picture of Hannah Alolia, who's now a professor in Finland, and also Kim Hassenkrug and Laura Myers, who are at NIH Rocky Mountain Labs. This funding was supported by the Fairbairn family, Kleberg Foundation, Barry Lyme Foundation, and I also have a disclosure that I am an advisor at Akari Bio, as well as my position at Stanford. So if you guys have any questions for me, I would love to take them.
I'm still muted. I'm saying if you have any questions, put them in the chat. I'm going to start. Thank you, Michal, for the nice talk. Do you think that because Mycobacterium tuberculosis is mimicking CD47 is what participate in its ability to be dormant?
So we absolutely think that the same as we see for cancer, where one of the first important steps is being able to coat themselves in the CD47 so that they don't get cleared by macrophages. We think it's also a really important step for the bacteria that are doing it and fungi that are doing it, because we think that what's going on here is that if you can coat yourself in CD47, then even if the macrophage can see that there's something there, it just ignores it, because SIRPalpha is telling it, is binding, and SIRPalpha is telling it, don't eat this, this is fine. So yes, we think it's a very important part in immune evasion, this first step of getting ignored by your phagocytes, like macrophages and neutrophils.
So have you tried to look at other bacteria or microorganisms that are known to be dormant in the cells, or parasites, for example? Have you screened a lot of microorganisms to look especially for CD47 micro, or not at all?
Yeah, so what we started by doing, and what was in that paper, is that we started by looking for host CD47. So at first we were looking with just antibodies against human CD47 or mouse CD47 and looking in these different infections. And that's what we published on. What we're working on now is actually using the CD1 as a molecular probe for mimics and screening lots of different pathogens. So at this point, we've screened multiple different bacteria and fungi. We haven't really screened very many parasites at all. If anybody is working on that and wants to work together, we would love to do that. But what we've seen from the fungi and what we've been seeing in general is that this seems to be a more important evasion mechanism for the smaller bugs that would normally be cleared by phagocytes. So a bug that's smaller than a single macrophage, what we see is that for things that are larger than a macrophage, they don't even bother with this evasion pathway. So I'm not necessarily thinking that the larger parasites would have it, but maybe intracellular parasites or like small parasites, maybe.
So if you, and then I'm going to read a question from Erica. So have you looked at intracellular bacteria such as the Rickettsia, Bartonella, all those tick-borne bacteria? Have you looked at those? Coxella burnetii?
For mimicry, no. And that would be really exciting to do. And we would love to do that and see if this is kind of a theme among tick-borne bacteria. That would definitely be very, very interesting, and something we would love to do. So yeah, that's a great idea.
So Erica is asking, CD47 is upregulated on cell types in LCMV infected mice that we don't typically think of being infected by LCMV. Does that mean that PEMs lead to something being secreted that leads to surrounded cells be upregulating CD47?
Absolutely. So it's a fantastic question. And that was one of the, so that was one of the main points that we were trying to get at with the different pathogen recognition receptor analogs that we use. So where I showed you where we trick the immune system into thinking it's infected and it's not, we use different types. And one of the types that we used was this, something that turns on TLR7. So this is something that immune cells would use when they're trying to sense if there is an infection, but they realize they're not themselves infected. So it's not triggering, you know, that they were infected on the inside, it's triggering that they were sampling things from the outside and found an infection. And so we know that immune cells that are seeing an infection are upregulating CD47 even when they're not themselves infected. We were also able to use to kind of tag infected cells versus non-infected cells, and we were absolutely able to see that a lot of bystanders, specifically immune cell bystanders, are upregulating CD47 in the absence of being themselves infected, but just because they're sensing this infection around them.
Okay. Any more questions? If you're typing, please raise your hand so I can know that you're typing. Yeah, Erica is typing.
So again, I'm so sorry for the late start.
It's
so
okay.
very badly about that.
It's okay. Okay, so yeah. So she added, does CD47 decrease on T cells after the peak of the T cell response to increase clearance of T cell to allow for T cell levels to decrease?
Yeah, so there's actually something very interesting going on with the T cells. Two things that I want to mention. So one thing is that T cells actually, during an infection, T cells go through this very early expansion, and then they go through a crash, right? So the T cell numbers, like the T cells get activated, they proliferate like crazy, they very much expand. At this point, T cells are upregulating SIRPa, and then when they crash, what happens is that they actually are losing CD47, and part of that crash in the population, part of that contraction phase of the T cell number going down is actually, part of it is them dying and part of it is macrophages eating them because of this reduction in CD47. So CD47 is actually also one of these things that helps us maintain our T cell numbers, and T cells use CD47 to kind of instruct macrophages, I should be here, I shouldn't be here. So that's just something that's going on just during homeostasis and is an important part of this expansion and contraction phase of the T cells and the immune response. One of the things that we followed is antigen-specific T cells and what they're doing with CD47. And we showed that actually in the paper. I didn't have the figure in here to show you, but within the antigen-specific T cells, we actually saw really interesting diversity. There was kind of a split always between the mice, where some of the antigen-specific T cells have really, really high CD47 expression, and some of them not so much. But there was definitely something going on there. And another side of that that was very interesting, that we published in a different paper, was actually that we also showed in the context of friend virus infection and Y10 mice, we showed that the antigen-specific T cells don't only upregulate CD47, they're also upregulating SIRPα, which was a huge surprise because people actually only really think about SIRPα on macrophages hematopoietic stem cells and neurons, and nobody had ever really thought about it on T cells before. But there we showed very clearly that it was coming up on a subset of T cells, and they were the most activated functional T cells. And they could even stay on during this chronic infection when the T cells are exhausted and highlight for us the T cells that were still functional. So there's something very interesting going on with the T cells, both from the CD47 side and from the SIRPα side. But I hope that answered your question.
Yeah, so she says yes.
Okay.
So Rachel is asking, have you looked at neutrophils and mechanism of uptake, for example, phagocytosis?
Yeah, so we have hardly looked at neutrophils at all, but they keep popping in to our data sets. So there is definitely something going on with the neutrophils. So when we do our in vitro system, we're specifically looking at macrophages and specifically looking at, Borrelia burgdorferi for the most part. But when we look in the mice, neutrophils keep popping in to the different immune cells that are really being affected by CD47 blockade very much. And we know that they highly express SIRPα, and we know that they're very important in bacterial clearance. So the neutrophils are absolutely involved here. And as much as I try to ignore them, I really won't be able too much longer because they just keep, there's absolutely something going on with the neutrophils. I have not really dived in yet what that is, but I at some point plan to dive back through these data sets that we've been collecting where the neutrophils just keep popping up. As a population that's really highly changed by 47 blockade, and try to figure out what is going on with them as well.
Okay, thank you. Is everyone else raising a question? Raise your hand if you are. Okay. 1 time, 2 times, 3 times. Okay, thank you, Michal. I think we're done with the questions. Thank you so much. It was very nice to have an immunology talk. It was the first one. I think we have a second one much later. Thank you so much again. I know that it must have been a bit brutal to just wake up and, and do that suddenly, but thank you so much for making it work. I don't know if you put your email somewhere in the presentation. If you haven't, I will just I usually upload. My
email is very easy if you're trying to contact me and have any more questions that you think about. It's M, which is the first letter of my name, and then Tal, my last name, T-A-L, M-Tal as one word, and then at stanford.edu, which is where I work. So do feel free to contact me if you have more questions, and I hope you learned something new. So.
Yeah, I think we all learned something new. I was actually saying to people, I had no idea what you're going to talk about. I was very intrigued about what the SIRPrises were, so I was very excited about your talk. Yeah, so thank you very much. And I was going to say I will upload your talk on YouTube. So, but you said that some of it was not published. So should I wait for it to be?
That's fine. No, go ahead and upload it.
Okay.
I'm very excited. I'm very excited about this, and I think it's more fun to just talk about it.
Okay.
So hopefully we'll get it published real soon.
Okay. Thank you so much. Thank you so much, everyone. And I'll see you all next week. Thank you, Michal.
Okay. Bye.
Bye, everyone.